Before we do that, I'm going to just have John Halamka make a few comments about goings-on at Mayo Clinic and what he's observed in his many forums over the last 2 weeks since we last met. John?
Well, thanks so much. And Brian and I were on a call a couple of days ago where we were asked to talk about the formation of the COVID-19 Healthcare Coalition. And FX, in case anyone wants to know, this coalition was formed at March 13th, 2020 at 6 in the morning. So here we are now 6 months into working together on what we thought would be an initiative that would last just weeks. And each week when we've got together for our various work groups, or now monthly for this meeting, we always reflect on where are we on the journey. And I highlight our usual stages that we go through, but if I reflect today on testing, contact tracing, and where we are on cures and vaccines, it goes something like this. We are seeing the emergence of rapid diagnostic testing, home-based testing, point-of-care testing. And so certainly the FDA has great interest right now in going from what has been this PCR test, initially using a very uncomfortable swab, then PCR using a more comfortable swab, serology, antigen. What is going to be the next few weeks? Well, maybe the ability to walk down to Walgreens and pick up a lateral flow assay, or pick up a simple test for $5 from Abbott and run that yourself. Or if not get a result yourself, get a result, take a photograph of it on your phone and get a result. So I think what we're going to see is just a lot of activity, especially as we think of back to life, back to work, back to school, an increased convenience and lower cost of testing. And obviously we just have to look at the AUCs. What's the sensitivity, specificity, recall, precision, and utility of those tests for a given purpose? Contact tracing, we've certainly seen a lot of states begin to think about it and some to deploy it. And I think a challenge with contact tracing has been adoption. If I said, download this app and people will be able to track your location and we promise it is private, is that something that most people would believe? And we know that there are protocols, the Google Apple Entity Notification Protocol, that are extraordinarily well engineered to be privacy preserving. But historically, that protocol existed without an overriding app, and each state would have to then choose a vendor or create an app. And you've seen now that Google and Apple are coming together to create a basic app. And so contact tracing, we hope, through education, through the use of easier to use and integrate apps, will take us from this period where we didn't have enough situational awareness about contact tracing, we didn't do it in an automated way, to more automation. We're going to hear today a really important discussion on where we are in the journey as we went from convalescent plasma transfusion, hyperimmune globulin, thinking of all the various products that will be derived, and we hope that this activity gets us as a bridge to where we are today with limited drugs in our armamentarium, dexamethasone, and vitamin D. I mean, there's lots of things being considered, to a reasonable approach to cure before we get to vaccine. And of course, all of us will be working together diligently as vaccines go through clinical trials to understand what vaccine should be used by what subpopulation with what efficacy, and how we'll get to a light at the end of this tunnel. And so many folks, I'm sure, have read of late things like Bill Gates' commentary or commentary from public health officials as to how long it will take to get to herd immunity. And I think we can just recognize one thing. We're all going to be in this together, and it will be a journey. And together, we will get to a better time. So with that, turn it back to you, FX, and look forward to the panel.
Thank you, John. I just want to remind everyone who's participating today that you can get more information in the chat, and please send your questions in as well. We'll be monitoring that so that we can bring those questions to the panel. Already in the chat, there are a couple of references for you to take in. One is a reference for a hyperimmune globulin trial that Dr. Mezzanotte will be referencing. And there's also a link to some documents from the FDA that relates to the emergency use authorization of convalescent plasma for your reading. Today we have Dr. Mark McClellan, our former FDA commissioner and currently the chair of the Duke-Margolis Center for Health Policy. Mark's going to be with us only for the first half hour or so. He's going to be our first speaker, and then I think we'll probably ask him a question or two, and then we'll proceed on with Dr. Nigel Paneth from MSU, who's one of the lead minds behind the large EAP registry study on convalescent plasma. Then we have a special guest, Mr. Nicholas King, who is a recent recipient of convalescent plasma, to give us his frontline experience. And then we've asked Dr. Bill Mezzanotte from CSL Behring, who is one of the leaders for hyperimmune globulin research in the country and already involved with a randomized trial. So without further ado, let's hear from Mark.
FX, thanks very much, and happy 6-month anniversary, I guess, to all of you. Maybe not that happy, but if we are going to be in this at 6 months, we really need groups like this one working together. I think it's going to take a combination of these approaches for a good deal of time to come. Better testing, better therapeutics, and a pathway to vaccine. And convalescent plasma products are a potentially important part of that. I'm going to talk a bit about convalescent plasma, but I think my focus today is really on providing some context about how FDA approaches issues around evidence. You know, why was there an EUA for convalescent plasma when there seemed to be some controversy about that? Does that have implications for, say, EUA for a vaccine? I'm going to talk through a bit of that and also some suggestions or potential ideas for maybe accelerating the progress on getting further evidence that clinicians, the public can use on how to make the most out of the convalescent plasma products, including hyperimmune globulin. So hopefully some useful context. Let's try the technical part first, where I try to share my screen. Let's see if this, can you all see a slide that says FDA approval versus emergency use authorization? Yeah. Okay, so far so good then. So just a reminder, I think people know this, but a product approval has a different evidentiary standard than an emergency use authorization. And it's also a less flexible standard. So product approval requires substantial evidence. It's based on totality of the evidence related to whether a product is safe and effective. And generally for drugs that we haven't used before, that means two adequate and well-controlled trial. It doesn't always mean that. It really depends on the circumstances of the particular product, the risks and benefits in context, and the strength of the evidence, but it is a pretty high bar. And I want to emphasize that emergency use authorization doesn't change that bar. It's you know, that the full approval is still there, but EUA was created in order to give the agency and everyone who's trying to respond to a public health emergency a more flexible approach to dealing with that emergency. The EUA was first authorized as part of Project BioShield, for those of you who were back there with me and others in the response to 9/11 and anthrax and the big concerns we had about bioterrorism and threats to America at that time from biologic agents. It was created in response to that, to create a flexible approach that could allow for use of a product in an emergency situation, maybe not as widely as it would be used if it was fully approved, maybe with some additional requirements for collecting additional data on top of the limitations on use, but meant to be flexible for the emergency circumstances at hand. So it is still a totality of the evidence standard, but it allows for at least some use where there may be a benefit, where the benefits outweigh the risks, and there's at least suggestive evidence in support of that. I just, looking at the actual EUA language, this may be effective standard comes in. By the way, I should say that this basic authorization approach has been modified over the years, modified in 2013 after experience with H1N1 pandemic to provide even more flexibility, but also some requirements for EUA use for the pandemic context. It's kind of written more specifically for that. And it's important to consider the context that we're talking about a threat that can cause life-threatening harm to individuals. And it does involve this risk-benefit analysis. And the challenge, as you know, in a pandemic is we don't have anything by definition that we know works. And it takes time and effort to develop the evidence needed to decide what's the best way to use therapeutics, emerging therapeutics, repurposed therapeutics, new therapeutics to address the pandemic. It's intended to support a flexible approach to that very challenging situation, like what we faced back around the time that this group formed in March of this year. So with that in mind, I want to emphasize that EUA can be used for specific populations, so maybe narrower than an approval. It can be used for different standards based on the context of use. And this is where I would like to spend a minute on convalescent plasma versus vaccines. For convalescent plasma, FDA authorized emergency use based largely on observational data analysis. So there wasn't, well, there's a very good Mayo registry compiled, and many of you all contributed to that. There was not a randomized study performed back in March and April when convalescent plasma, which is available for use and increasingly available as more people develop and recover from COVID-19, it's out there. So it started to be used, and the reason FDA concluded that it met the standard, and I think this is an appropriate conclusion, was that we had data, thanks in part to the registry, and good part of the registry, that the product appeared to be safe in severely ill COVID patients, that is those who were hospitalized. We've used it before in many other contexts and generally had no significant safety issues arise. There were questions about how effective the treatment was, and you remember Francis Collins's comments and so forth. But as Francis stated after the fact, after all that controversy, it still met the standard. He agreed that it still met the standard for EUA, that it may be effective. It was not harmful in this population. And it's not to say that's the end of the line. FDA would very much like to see, and all of us would like to see, more compelling evidence developed on benefits versus risk. But in that population, for that use, based on the safety profile, it met the standard. Vaccines are very different. And for those who are interested in this topic, Scott Gottlieb and I had an editorial earlier, the op-ed earlier this week in the Wall Street Journal about how EUA is likely to be applied in that setting. And I think there's a 99% chance that when a vaccine starts to be used, it's going to be through an EUA, but it will be based on a very different set of steps because of the different context for use. So this is a product that will be given to potentially millions of people, many of whom are healthy or certainly don't have any severe life-threatening illness at the moment, but who do face some risk, varying risk of COVID. And this is exactly what the EUA was designed for, that flexibility that I described. So what we think is likely to happen, and I think the experts at CBER, the biologics center that are overseeing all this, think is likely to happen, is that you'll see a staged access to the vaccine. First thing that has to happen is that the trials that are underway now, the big randomized clinical trials, well-designed, are going to have to demonstrate that they reach an endpoint. So those aren't even going to have a chance of concluding until the data safety monitoring board for those studies see whether or not, after enough events related to COVID infections and severity of infection have occurred, to power a comparison about whether the vaccine is at least 50% effective in reducing the number and severity of events, meaning like whether you get COVID infected with symptoms at all, or whether you go on to hospitalization or other severe consequences, all spelled out in guidance from the FDA. After that happens, a company can provide an emergency use authorization to the FDA, because it does take months to do a full authorization. And it is likely that emergency authorization will focus not on the whole population, but on a subset of the population that is really likely to benefit from the vaccine. So it's not going to be about herd immunity at that stage so much as it is about basically a form of therapeutic for social workers, healthcare workers, depending on the results of the early studies, maybe some nursing home residents or other people in the high-risk categories, but a limited use authorization coupled with substantial additional data collection, both further collection of the safety data sets on the 30,000 or 15,000 people at least that are going to be randomized to getting a vaccine, and we'll have months of safety data already, at least a couple of months, and more time to follow them while this limited emergency use occurs, and also an opportunity to do data collection, registries, or equivalent to registries using real world data on potentially thousands, hundreds of thousands, more individuals during the EUA phase for the first vaccines that get into use. But that's only going to be after FDA has reviewed the evidence. FDA is committed to a public meeting of their vaccine advisory committee, in which FDA and the company will present their conclusions about the data they've seen, and we'll have a chance for independent experts to take another crack at this. This is after the DSMB has done its review, and a chance for the public to get more of an understanding of just how good the evidence is and how far it takes us. And then even if it is authorized for emergency use, I expect the CDC Immunization Practices Advisory Committee will also weigh in. There'll be more chances for public expert input on how exactly this stage process access should go forward. All of that, a very different approach for convalescent plasma from vaccine, all that can happen under the emergency use authorization framework, which again is intended to grade access to the development or the availability of evidence and the totality of evidence on risks and benefits. So you can think of let me move on to talking a little bit about how we make that evidence better, and then I'm going to come back and pull all this together, hopefully really quickly. So where are we in terms of generating evidence? Well, it would be great to do randomized clinical trials. That is hard to do in a pandemic where the site of infections is shifting, and where, at least earlier on, there were a lot of hospitals that were really hard hit and didn't have a whole lot of time to spend entering data and setting up clinical trials. We've seen a lot of clinical trials be started in the US and around the world, literally thousands. My guess is that only 10% of those or less are actually going to yield meaningful results that will impact clinical practice, because when you set up a trial quickly and you're just doing it at one or few institutions, you may have issues in study design that may make it difficult to generalize to other settings and patients. And you may also have difficulty in reaching power calculations. I can't tell you how many studies have ended up stopping enrollment before they reached the point they were designed for sufficient power, because the epidemic went away in the particular region where that study was being performed. That's why trial networks are important, and that's why the work that Active, supported by Operation Warp Speed, is doing is so critical to identify ways, whole sets of hospitals and health systems that can work together, both the inpatient and outpatient setting, to get more compelling evidence that gets past that those kinds of problems on at least the most important therapeutics where we don't have evidence now. The challenge with that is that it's awfully hard to set up. There are only a minority of health systems and healthcare organizations participating in Active for only a minority of their patients, because it is a lot of work through the data collection, the enrollment, the site management, and so forth for a really good NIH standard trial network. That's why some of you have heard me and colleagues that we work with, like Rob Califf, Brian Anderson, and others, talk about the potential value of a practical trial network, something that could be designed to augment these other activities. I see there's a missing spot on my slide there. And potentially provide some additional support for evidence. So what would this look like? How do you build a practical trial network quickly in a pandemic? Well, we've got some models to use. The recovery trial, some of you may be familiar with, in England, has enrolled tens of thousands of patients by keeping it simple. Building a limited amount of additional data collection into the real world practice of hospitals all across England, focusing on just a few key outcomes, a limited amount of data collection, a practical approach to randomization that does not include placebo groups or double blinding. And there are versions of this trial being implemented in networks in the United States now. The iSpy COVID network, led by Laura Esserman, is doing kind of a simple version of phase 2 studies, particularly on ICU patients, but severely ill patients. The REMAP CAP network, led by Derek Angus at UPMC, it's kind of following the recovery network model, limited amount of data collection. What we're trying to do with those groups is give them some additional resources and support to make it easier for more sites to participate in this. Again, this is not a replacement for active and those intensive trial networks. It's meant to recognize the fact that if you look at the active priority list for compounds that they'd like to test, it's much longer than active has capacity to undertake now. So key features here are simple protocol, integrates with clinical care as much as possible, limited number of key endpoints like an ordinal scale or diary for hospitalized patients, the severity of COVID, the COVID case, are they needing intubation, et cetera, whether or not they need ventilation assistance, or simple few other drugs, but limited with an easy data collection process that makes it as minimal effort as possible for these busy clinicians who are caring for patients, especially in the hospital, to enter the data needed to support the trial. Leveraging existing networks here. So iSpy COVID was built off of iSpy, and it's applying the same principles that were used in kind of these simpler approaches to trials for severely ill cancer patients to severely ill COVID patients. REMAP CAP was based off of a clinical, a community-acquired pneumonia network, a respiratory trial network, and keeping it simple for complex patients by keeping the cost down to make it easier for sites to participate. But we're also working on providing tools and assistance for the sites and focusing on, again, practical questions that are not, we don't have the bandwidth to answer through the traditional trial networks. And it's just to add the other source of evidence, very important to get to that totality of evidence that I talked about before, is real world data sources and data elements. And here, good model from convalescent plasma, where there's a lot of effort to capture data on as comprehensive a population as possible, attention to collecting data with some rigor so that differences in data quality and questions about fit for purpose of the data that's being collected could be addressed, an infrastructure to put this together, thoughtful analysis, and combination of resources. I understand now there's some steps underway to try to pool some of the clinical trials, those individual clinical trials that maybe weren't well powered enough or weren't exactly designed in a consistent way, to try to add to this evidence as well. But again, this is very important for contributing to the totality of the evidence. So, what might be ideal here, and maybe how could further progress on convalescent plasma is about better. So what active is doing, any sites and set up well-designed trials that meet FDA standards, that's just critically important. But it is very likely to be capacity constrained in a pandemic. And that's certainly what we've seen in this one, where very good aside from monoclonal neutralizing antibodies and some of the anticoagulants and other drugs that could be quite helpful. Importantly, in reflecting those EUA principles, a huge amount of effort has been going into getting the randomized trials set up effectively for vaccines, where, again, it's hard to see a path to use even on an emergency limited basis until there is that randomized trial level evidence on hard endpoints demonstrating that the treatment is that the vaccine is safe and effective, and then building on that with additional real world evidence. But this is where a practical trial network can come in. Short list, potentially some phase 2 studies, smaller studies to detect a signal for benefit. on treatments where we already have some safety data, either because it's generic products that are already available, or maybe they're brand name products, but approved for other uses. So there's, again, a good safety profile. And then phase 3 studies for compounds that, again, can't make it into active, or questions that can't make it into active, but they're good to answer. So products in hospital that could reduce mortality. That's where recovery in England has made such a big difference around compounds like dexamethasone, and as well as negative findings on compounds like hydroxychloroquine. And then other questions that I think will be increasingly important in the months ahead: treatment combinations. You know, for some people, are multiple with the vaccine for at-risk questions of illness. Maybe there are still, on how those go, there may be some questions here that a practical network could answer. Timing, for example, extending to other patient populations where it may be feasible with an EUA in place to still do randomized studies. But certainly for treatments like hyperimmune globulin, there's a well-designed trial getting underway now about in a minute. But augmenting that fast into the pandemic is really important. But acting thoughtfully on following some best practices and some foundations for developing better evidence faster can also make a difference.
A lot of ground quickly, and it's because you've had such good command of it, and just want to thank you for all the service you've had since being FDA commissioner. You've stayed in the game and are creating new relationships, new organizations, and it's exactly what we need in this time. So just really happy that you're
Nice.
with us and helping the country to get through this. We know you have to be leaving.
Actually got till about 3:05. If
okay.
I want to hear what Bill has to say too. So
okay, all right. All right, Nigel, I think we'll go right to you if you want to show your slides. And
yeah, here we go. Okay, I think it's gonna move upwards.
That's good.
Sorry.
Thank you. We can.
All right, we're in business. Okay, I'd like to give you a very brief overview of the effectiveness of convalescent plasma as demonstrated or not demonstrated by the studies we have available to us. I'm Nigel Paneth. I'm from Michigan State University. I'm part of the COVID-19 Convalescent Plasma Project, a group of academics who organized spontaneously in March to begin thinking about how we might study convalescent plasma in this situation. I'd like to give you an overview of the evidence, starting with safety issues first, and then discuss 3 forms of evidence of effectiveness and accelerating degrees of confidence which we can place in the evidence. So I'll start with the treatment control comparison studies, which were not randomized, but which clinicians who treated patients with convalescent plasma compared them to other patients who did not receive convalescent plasma with a range of degree randomized trials that are out there and in the public domain, and then putting it ahead of randomized trials in terms of its importance, the impact of the studies of antibody level in plasma. This is not to say that I think that in general randomized trials are not the ultimate source of information about the effectiveness of treatment. I'm just dealing with the randomized trials available to us at the moment. So on safety issues, it's important to keep in mind that something like 4 to 5 million units of plasma are used in the US annually, mostly in the form of fresh frozen plasma or so-called 24-hour plasma. And convalescent plasma differs from usual plasma only in that it comes from people who have been infected with COVID-19 and have recovered and have been asymptomatic for a minimum of 14 days, often 21 or 28 days. It doesn't, but it's not prolonged in COVID-19. And so we don't think people are likely to be infected with COVID-19 from convalescent plasma. The analysis undertaken, as Mark referred to, the EAP database, which is now much larger than this, but on the first 20,000 patients, I was happy to participate in the analysis that looked at the complication rates and the two feared complications of plasma administration: TRALI, transfusion-related acute lung injury, and TACO, transfusion-associated cardiac overload, occurred in very few patients, far less than 1%. Other allergic reactions were also very rare, and any form of severe adverse event occurred in fewer than 1%. So everything points to not to worry excessively about the effectiveness. Now, we were able to identify 7 studies done outside of the US. Plasma were compared to untreated patients. And you see here that in every one of the 7 studies, the mortality rate was lower in the patients who received convalescent plasma, sometimes markedly so, as in the study from Iraq, sometimes not so dramatically so, as in a study by Zhang from China. But overall, if you look at all the studies combined, and I pulled them here in a very simple manner, excluding a couple of studies that could not be pulled for a variety of reasons, the net picture of the effectiveness from these admittedly observational studies was about a 60% reduction in mortality. Now we have 5 studies that were done in the United States from a range of locations in the United States. And they are, they show also a range of findings, and I have to minimize my screen here so I don't hide what I'm saying. Okay, but again, all but one study showed a reduction in mortality. The first 3 studies that we know of, the only one that's been published is the study by Lee from Wuhan, published in JAMA in June. The other papers are on the Medical Archives server. The problem that was noted by Mark was the termination of trials too soon. This happened in Wuhan because cases dried up. The Dutch trial was stopped for reasons that are not a little more complicated than this. The Spanish trial was stopped because patients dried up. If you look at those first 3 trials together, they individually, they're not statistically significant in terms of the decline in mortality, but collectively they are. But only this past week, a 4th trial emerged from India, a larger trial, considerably larger than the others. It did not show a mortality reduction. I won't make a deep dive into what was going on in all the different trials, but I will say that one feature of the trial from India was that the level of antibody in the plasma use was not as high as it was in the other trials. For example, the Spanish trial required a high antibody titer before transfusion, and both the Dutch and the Chinese studies were able to demonstrate relatively high antibody levels in most units transfused. If you pool the first 3 studies, you get a result quite similar to the observational studies of about a reduction in half in mortality. Even if you include Agarwal from India, you still have a reduction in mortality, but not quite as notable. Now, the antibody findings from the Mayo Clinic will, I'm sure, generate much discussion. The paper is on the medRxiv server, and these are taken from that paper. of the plasma, because blood banks often keep those. At least 45 blood banks were involved in this effort. Have that plasma sent to the Mayo Clinic, where it was tested for its antibody level by several different tests. And this test is based on gamma globulin from the Ortho platform. And and as you can imagine, no one knew at the time of treatment what the antibody level in the plasma was. And to some extent, this can be viewed as a quasi-plasma was just unknown to anybody at the time. By the way, low, middle, and high are set by percentiles, the 20th and the 80th percentiles of the antibody titer. So high means in the top 20%, low in the bottom of the base. We do see a reduction in mortality, a gradient as you go from day mortality rates. If you focus on people treated within 3 days, we see an even stronger signal of efficacy. A small Israeli study just published or just presented has found a relative risk of mortality, really also quite similar to this, about a halving of mortality. 4 multivariate approaches using 3 different methods. So we actually have as many as 12 different estimates of efficacy. And not every one of them is statistically significant. Some are and some are not. But the general tendency of all these analyses is in the same direction, with about between a 20 and 30% reduction in mortality overall, and a higher than that in the people treated early. Another way of treatment, and focus only on the people who are not that advanced in illness, we do see that strong signal of efficacy, but not in the people who are ventilated baseline. So finally, conclusions. Even absent a large randomized trial, which we all wish we had, it's hard not to see powerful evidence of convalescent plasma efficacy in reducing mortality in the various forms of data available to us. I should say that there are several trials in progress in the United States, most notably in New York City at Montefiore, where well over 200 patients have been enrolled. They're expanding to sites in Florida and Texas. There are other studies going on in other parts of the country. They're going to try to compile them together. In fact, they call their process of putting them together compile. So hopefully we will see the results of US randomized trials soon. No US randomized trial has been published as yet. I think the data showing the dose-response relationship, which was unknown at the time of treatment, is hard to explain away. It's not easy to think of confounders of that relationship. I think it was this information that was ultimately most convincing to the FDA in making its announcement on August the 23rd of the expanded use agreement. And I so appreciate Mark's explaining the distinction between emergency use authorization and a more permanent form of endorsement, such as is usually available. It's also very clear that early treatment is better than waiting until the disease is more severe and that complications have ensued. That's clear in almost every database that we've looked at, including the Mayo Clinic. And so I think I, like Mark, agree that the FDA did the sensible thing. There was evidence. It wasn't perfect evidence. Evidence rarely is, but it was strong evidence. And I'm happy to share these slides with everybody. And if you need the references to the papers, we can get you those as well. So thanks very much to listening, and thanks for inviting me to this very important meeting.
Thanks, Nigel. And that was really a great summary of this really, really complex area. So we now are going to turn to a special guest, Mr. Nick King, who was a recipient of plasma. I wonder, Nick, could you turn your camera on? Great. Well, thanks. And I'm going to ask Nick to tell us a little bit about what he went through as a patient at Johns Hopkins during the month of July.
Okay. Hello, everyone. just a little backstory. I am a kidney transplant recipient, polycystic kidney disease, transplant in November of 2004, dialysis for 3 months prior. I had a double nephrectomy in October, and then a kidney transplant November 1st of 2004. Since that time, I've been, what I'll consider healthy, do my blood work once a month, and see my nephrologist annually. On July 18th of this year, I started to exhibit symptoms of what I thought was COVID, what I was hoping was not COVID, but I called my nephrologist. I had coughing, wheezing, a fever of about 101, headache, body aches, chills, etc. He wanted me to get to the ER at Hopkins as quickly as I could. So that's about a 2-hour drive from where I was staying at the time. I got there Saturday afternoon around 1 o'clock. They did a COVID test for me, came back positive, and admitted me at 4 o'clock. They put me in a negative pressure room with all of the the nurses taking the appropriate precautions. I think I was approached about the plasma by the infectious disease expert for the transplant division of Hopkins, and that was probably Sunday morning, where they came in and said, we'd like to talk to you about convalescent plasma. They explained to me, the pros and cons, that the risk was limited, but there was some risk of allergic reaction, which you folks all hit on. But I was, very agreeable to take the plasma, because at that point, I didn't know how I was going to progress with COVID being immunosuppressed. I had to go through the process with the person at Hopkins who I had to sign off on it, I guess, or was running the program. He was actually at home because he had tested positive for COVID, and he had taken the plasma himself. So we had an interesting conversation about that. But after agreeing that I was a candidate and that I wanted to do it, it probably took 6 hours for them to get the appropriate approvals. I was given the plasma about 3 o'clock in the morning on Sunday, the 19th, and I was actually discharged from Hopkins on Monday afternoon around 4 o'clock. And for me, it's been, an extremely good process. I was probably symptom-free within 10 days. I was actually feeling very, very good. Immediately after I left the hospital, I was able to walk a mile or two, and then I probably 3 or 4 days later had a little setback with symptoms that came back to kind of how they were originally, and then over the next 4 or 5 days, I was back to symptom-free, and like I said, day 10 or 11, I was symptom-free and have been symptom-free since. So that's my story.
Nick, thank you for sharing that, and I know you've been involved with other clinical trials over the years, so thanks to all your participation in clinical science. Thanks for sharing your story. And you're actually, now that we see what the evidence that Nigel had just shown us, we're seeing that early treatment is looking to be the best option for patients. And so you actually met those criteria and got very efficient care at Johns Hopkins. So thanks for sharing that, and we'll have some questions for you during the panel. We'll go now to Dr. Bill Mazzanotti, and Bill's going to share us a few slides and talk about the study he's involved with now regarding hyperimmune globulin and other therapies. Bill.
Yeah, thanks, FX, and thanks to Nick for telling his story, because the whole reason we are on these calls is for patients like Nick, and so it's so great and satisfying to see him have recovered and to hear his story. So I represent one of the plasma manufacturers, and we create hyperimmune therapy, which I'll get to in a minute, but I'm from CSL Behring, and this comes full round because CSL Behring in part is derives from Emil von Behring, the first Nobel Prize winner for using serum for the treatment of diphtheria right around the turn of the 20th century. So let me just bring up my slide, and just to remind some of the audience who may not be quite as familiar, a hyperimmune is somewhat different than a convalescent for transfusion, but it's the same at the same time. So it's derived from the same gift, which is convalescent plasma given from donors who donate their plasma. For transfusion, of course, we've heard a lot about it, but people are giving it back relatively immediately, like in Nick's case, which is great because timeliness is really important in this disease, as you just heard. The challenge a bit is that the levels of the antibodies in the plasma can vary from donor to donor, and you saw in Nigel Paneth's slides, the levels of antibodies really do make a difference. And the other challenge, of course, is that normally we give plasma not for the immune globulins immediately, but we've been given plasma for all the other proteins that are available in the plasma. And you get those proteins as well when you directly transfuse them. And so we have to be cautious that that doesn't cause its own reaction. In the hyperimmune, we take pooled plasma from many convalescent patients and make a large batch. It's sent to a manufacturing facility where it's pooled and processed, purified. Virus is inactivated later in the process versus upfront in the convalescent plasma for transfusion process. And it's purified, meaning all the other proteins are taken away. So it's essentially just the immunoglobulin you're getting. And here it's mostly IgG. IgM is processed out as well, which may be actually a slight downside to the hyperimmune versus the immediate convalescent plasma. And what this is then, you essentially create a medicine that can be stored, it can be shipped, and it can be reconstituted at the whim of the treating physician. So we have known about the use of convalescent plasma for a while, and have known that it would be effective because, well, we didn't know convalescent plasma would be effective, but a hyperimmune is effective because all of the manufacturers have some experience in making hyperimmunes. And in fact, tetanus and rabies are diseases that are often treated by hyperimmune every day around the world. So we created what's called the COVID-19 Plasma Alliance, which is an alliance of about 10 manufacturers. And we're working together to collect plasma from each of our partners in whatever regions we have collection centers, talk to the health authorities. We are then doing analytics on those collections together using a common neutralization assay, which is, of course, one of the challenges in this whole field is the lack of a standard neutralization assay. And we saw that rear its ugly head when we looked at the FDA approval for the convalescent plasma for transfusion and the single test approved. We've already made small scale batches for a clinical trial made by Takeda and ourselves, CSL. But other commercial batches are underway at much larger scale. So we have over 1,000 liters for the clinical trial, but we will have over 10 to 20,000 liters for commercial use immediately, and then ultimately much more afterwards. And the clinical trial is then going to be underway, and I'll talk about that a little bit in the next slide. But it will include this alliance plus 2 other manufacturers, Grifols and Emergent, and therefore almost all of the plasma manufacturers of relevance are participating in this single trial. So we will have consistency of the results, and hopefully we'll avoid some of the challenge that you've seen with Nigel's data, which is conflicting data from different sources. And then we are planning to file for emergency use authorization in the US and conditional marketing authorization in Europe. And they're essentially very much the same flavor of approval as Mark so eloquently talked about earlier. So this is our trial, and it's called Insight 013. Very exciting title. And I sent the link in the chat room. So this is up on clinicaltrials.gov now. So we're going to be screening 500, randomizing 500 patients in a 1-to-1 ratio to either the hyperimmune globulin plus standard of care. And what's standard of care? Well, it is remdesivir at this point. And you did see, interestingly, the NIH feel that plasma for transfusion did not meet the level of standard of care, which is good news for our trial, because I guess we would have had to incorporate that into the trial as well. And then it's placebo plus remdesivir as the comparator. And we are using the similar ordinal scale to show efficacy, so people improve at levels of improvement compared to placebo, and looking for a significant difference. The sponsor of the trial, the sponsor of the trial is the University of Minnesota. We are using the NIH's Insight Network, which is around the world and did do the remdesivir trial. And the NIH is the holder of the IND and filing for the clinical trial approvals in Europe as well. So that trial is to start imminently, and we hope once it starts, the total time to the endpoint is only 28 days. So if we enroll rapidly, we should know data rapidly, and we're very hopeful we'll know data at the end of the year, which would be a really important step. And then we would go, depending on the results, of course, assuming they're positive, we would file for emergency use authorization. And we have had discussions with both Europe and US regulators in advance, making sure their needs are met. I would say a couple of things. One, Mark mentioned his large simple trials. And I would think that if we are able to demonstrate efficacy, a nice way to expand understanding of where else these therapies might work, or in what combination they might work, would be in trials such that Mark has described, because I think what we really need to get to next, now that we're starting, as FX said earlier, starting to accumulate therapies that are working, we have to figure out how they work together. And I think even before a vaccine gets to the market, we have to know how these therapies that reduce morbidity, mortality work together, so we can take the fear out of society and allow us to get back to some sort of normal life. So I'm excited to get the trial started. I'm excited to see the results. They call it an experiment for a reason. I don't know what the results are going to be, but we're very hopeful and very enthusiastic about adding to the armamentarium of treatments for physicians and for patients. So FX, thank you for inviting me, and I'm happy to take questions.
Thanks, Bill. I'll ask Nick and Nigel and Mark and Brian Anderson to turn on their cameras if they're all still present. And, there's just so much we could talk about, but what's clear, I think, is this idea of collaboration. And so I know each of you has been involved with clinical research for many years, but I just wanted to hear a little bit about, Mark described potential for putting this together, and just wondered if we could hear your thoughts about, where we are in the pandemic now, and what the next 6 months looks like. What do you think what we will accomplish? And maybe I'll ask Brian Anderson to talk a little bit about that. He's been very much involved with the Coalition for Plasma Collection, as well as Coalition for Plasma Analysis.
Sure, I'm happy to let Mark go first if he's still on FX.
Is he on? Yeah, he didn't turn his
camera on. I don't know if he is. If not, I'll go ahead and go. So, I mean, I think when I think of what the next 6 months look like, FX, part of what I appreciate is the need to, redouble our efforts to base our decisions in science. Bill said something earlier this morning that really resonated with me, which is, during a pandemic, there's a sense of urgency to act and act in ways that are decisive and quick. And those may not always be, necessarily ways that lead to robust science, that allow us to have the most informed decision making possible. As I look to the next 6 months, I see groups like those that we have here, the COVID Alliance, the Fight Is In Us coalition of coalitions, and others have the opportunity to more intentionally ask ourselves how we can support science, randomized controlled trials. Dr. Claire Pomeroy has been advocating for, and I really agree with this idea of a national coordinated effort to coordinate the enrollment of patients across trials as another idea. How can we come together in a more coordinated fashion to support science as a real opportunity in the next 6 months for coalitions just like this, FX?
Great. Nigel, I wonder if you could comment upon the work you're doing to bring hospitals into a data sharing alliance, maybe hospitals that never participated before. How hard is that to do in the middle of a pandemic, and what are we learning from that?
Well, everything that's hard to do ordinarily, which is everybody to play in the sandbox together, is doubly hard during a pandemic. We have had a lot of interest from hospitals. What we're doing now, I think what FX is referring to, is the fact that the hospitals have done a lot of transfusing. We're trying to get data from them about their non-transfused patients as well, match them carefully, and try to do an analysis of essentially the Mayo case list roster of treated patients to untreated patients in the same institutions. That's steadily mounting, getting stronger. As you know, FX, you're very involved in it. We discuss how to do it on a weekly basis, and we're getting closer to that. But there is I think we're getting closer to having that kind of an analysis, which will be supplement what I've shown you already, and hopefully will again inform us, even though it's not a randomized trial until the RCTs come out, which I hope will be in the next couple of months. As far as the next 6 months, how close it will look like to the past 6 months, will it look like the past 6 months or the 6 months that preceded it? Which will it resemble most? To be perfectly frank, I think it'll resemble most the past 6 months. I don't think we're there yet. I don't think we're there yet as a country. I don't think we're there yet in terms of masking and distancing. I think if individuals began to really take this as seriously as it ought to be taken, if all these if statements, if we get a good vaccine soon, if testing becomes simpler, as was being discussed by John Halamka, simpler and more available, if we could test people much more frequently and regularly, then I think that could change. Those things have yet to be demonstrated. And that's why I'm afraid I think we have to gear ourselves for a long winter.
And Bill, I know you've been very involved with the team at the Fight Is In Us to help promote more donation. Can you give us a few ideas about the best sources of plasma? I know this is something you've been giving a lot of thought to.
Yeah, I just want to mention one thing. I might be a little more optimistic than Nigel. I think the next 6 months are, I'm hopeful. I think that some of these things that he said, I'm more of a when guy, and believe that some of these treatments are coming. I'm not sure a vaccine's coming, but I think, there's a lot of studies out there and I think all it has to hit is a few of them for us to have a lot more tools in the toolbox of the treating physicians. Convalescent plasma is a precious source, and we need to get it from the patients that have had COVID. And this has been a known challenge. It's actually one of the reasons we made the alliance to begin with, because we knew we all knew how to make a hyperimmune, but we all knew that getting the plasma was going to be the trickiest part. And it's proven exactly what we thought, which is identifying, motivating, reinforcing those donors is not easy. And of course, these same people have just been, you heard Nick King's story. I mean, he was through a medical catastrophe of his own, and now he has to come back regularly and be and be implemented all over, instrumented all over again. And that's not easy to do over and over again. So we have to figure out a way to support these people, motivate them. But what I really want to find, and this is why testing is so important, I want to find those asymptomatic carriers, because they haven't been through what Nick's been through, but they still have the antibodies themselves. And I think they are the secret to expanding the pool of convalescent plasma.
So maybe it's going to be those college students who are getting infected this semester who could really contribute.
God bless them, FX, you know? That's right.
Well, I think we continue to need to spread the word about people like Nick who've had successful treatment. And I think encouraging donation of plasma and awareness of it among the whole population is really key. I'm going to give the last comment to Nick if he wants to say a parting word for us. We want to thank you again, Nick, for your participation in the research, and congratulate you on your recovery as well.
Well, thank you. Well, I'd just like to say, look, I'm thankful to all of you that are, working hard to come up with some solutions to this problem. I've been the beneficiary of with my kidney transplant and all the immunosuppressant therapy I've had, and I'm leading a pretty normal life, given the fact that I had kidney failure at 39 years old. So, to all of you, I thank you for that. I'm actually doing a study now that you guys may be aware of through Hopkins. They're actually testing the level of antibodies in my blood for the next 6 months, every 2 months for 6 months. And I just feel it's, almost my obligation to participate in these studies where I can, because I've been the beneficiary of such, medical progress. So thank you guys for all you do. And I'm happy to participate.
Well, thank you. And thanks to all the panelists for all you do every day. Thanks for the audience for participating and for all the work we are doing in union to shorten the pandemic. Next month, we will again be on the third Wednesday at 2:30, and we will be bringing you a panel discussion that relates to back to school and some of the challenges and lessons learned already. So stay well and stay safe. Thank you.
Thank you very much.
Thank you, everybody.